Good day. My name is Vangelis Daskalakis from the Department of Chemical Engineering at the Cyprus University of Technology. My talk will be on the mechanics of photoprotection in the major antenna photosystem 2, which was a SuperMUC project codenamed CD Dynamics LHC2, which was also awarded 17 million standard CPU core hours. Research in the lab is based on bioenergetics and the oxygen cycle. We have photosynthesis and the oxygen evolution, as well as cellular respiration, where oxygen gets reduced to water from humans. And this closes the cycle, the oxygen cycle. Photosynthesis and cellular respiration rely on protein complexes to harvest and convert energy. So natural macroscopic processes can be explained by atomic scale details and have useful applications. Structural details are the basic elements to understand nature, as well as protein structures. However, not all information is stored within a structure. So to go from an initial conformation to a final conformation, we need also the dynamics, which means the function of the protein. So protein structure and function are related to the motion of protein atoms on free energy surfaces like these between energy minima, the red areas here. So we have flexible proteins with many energy minima on the free energy surfaces and unrestricted atomic motions, as well as rigid proteins and fewer energy minima with restricted protein motions. Our methodology, our approach is based on molecular dynamics, a simple theoretical basis to study these motions of proteins. If a mass m is at position x0 and a force is applied on this mass, then the mass is moving to a final position x1, obtaining velocity v over time delta t. This motion is based on Newtonian equations of motion. And this is how we also move atoms in proteins. So, given an initial structure from crystallography or predicted, we can recover a free energy surface, and we can go from one conformation of the protein to another conformation by moving its atoms. Given a force F on the atoms, given the potential energy surface and the mass of the atoms, we can have the trajectory of atoms, their positions over time. So we can observe the protein that functions like we would observe it under a powerful microscope. So, as I told you, photosynthesis and cellular respiration is the main focus in my lab, and these rely on protein complexes to harvest and convert energy. Photosynthesis relies on photosystem 2, which converts, oxidizes water to oxygen, and cytochrome C oxidase, which reduces oxygen back to water. These are also coupled to anabolic and catabolic pathways to harvest energy. Our research for the last 6 years is based on structural details of that conversion, which are missing for photosynthesis, and how we can get from a conformation 1 to a conformation 2 for certain proteins of photosystem 2 that are related to this energy conversion. In photosynthesis, actually, a small introduction, a complicated story evolves antennas or light harvesting complexes around photosystem 2, as you can see here, absorb sunlight under normal light conditions and direct it to the reaction centers of the photosystem 2 to be converted into chemical energy. However, in bright light, the absorbed excess energy is not reaching the reaction centers in order for them to be protected, and it is dissipated as heat and lost. So we have two conformations, two states of the light harvesting complexes or antenna of photosystem 2. One is the light harvesting state, which absorbs the light under normal conditions and directs it to the reaction centers, and one called photoprotective state, which dissipates the energy, the excess energy from the sun as heat. No crystal structure yet exists for the light harvesting conformation, and this is what we're looking for. We're interested in this structure, and there is a global interest for that structure because it can lead to the development of artificial photosynthetic systems, as well as tolerant plants and the increase of biomass under harsh conditions of soil, water, and temperature. Especially for the latter, we know that the biomass production under photoprotection, under excess light, is severely reduced as the and a lot of this energy is dissipated as heat and lost, whereas in normal conditions, the biomass production is increased. So we can check this mechanism, the photoprotective mechanism, if we know the photoprotective or the light harvesting structure, and direct these energies to biomass production. But first, we need to resolve the light harvesting conformation of the antenna. In vivo, the transition between light harvesting and photoprotective states is achieved by the interaction of the light harvesting complex or antenna with a protein called PSBS, which induces protonations at the luminal space side of this protein under a pH gradient across the thylakoid membranes of 1 to 1.5. And this all leads to aggregation of light harvesting complexes and the dissipation of energy as heat. However, molecular details are unknown, and that's what we're looking for. So we know the photoprotective state, there is a crystal structure for that, and we're missing the light harvesting state. Our model is the light harvesting trimer, the major antenna of the photosystem 2, which is a protein scaffold embedding carotenoids like neoxanthin, lutein, violaxanthin, and chlorophylls, as you can see here. Also, we have predicted the accurate position of PSBS around the LHC2 antenna of photosystem 2, which is this green protein here. And we have embedded this antenna in thylakoid membranes, as you can see here, or liposomes, in the presence and the absence of PSBS protein. We monitored the aggregation, the antenna aggregation in the absence and the presence of PSBS protein. PSBS is this purple here, and the antenna are the green proteins here, as we watch this process on a microscope. And we found out that the antenna shape is changing, moving from the light harvesting to the photoprotective state. And different thylakoid lipids are accumulating around the antenna of photosystem 2 at different states of pH and the presence or absence of PSBS. These are the details of the shape change of the antenna, moving from the light harvesting to the dissipating state. There is a delicate interplay between two thylakoid lipids, like the diacylglycerol lipid, and this monogalactosyl diacylglycerol lipid. These digalactosyl diacylglycerol lipids are accumulating at the luminal space, where this lipid is expelled and moving to the stromal space. And this movement leads to the aggregation of the antenna in the presence of PSBS. Changes in the antenna shape also induce changes in the thickness of the thylakoid membrane under photoprotection, and there is also experimental evidence for this and the confirmation. The sampling for this study was at 150 microseconds total, whereas the state of the art of the literature at the time was only 14 microseconds. So we were able to identify the light harvesting conformation, the green cartoons here, and how it is converted to the photoprotective state. The arrows show the way. in a thylakoid membrane. This is a movie of the transition of one monomer of the LHC2, the antenna trimer. The thickness of the thylakoid membrane can be also controlled by sterols, as we found out. So we can induce the same conformational changes on the antenna by introducing sterols within the thylakoid membrane. Now, based on the literature and our findings, we have proposed a mechanism for this transition between the light harvesting and the dissipating states under normal pH gradient in the thylakoid membrane. Normal pH gradient in the thylakoid membrane, and in the presence of PSBS, we have protonations at the luminal side of the trimer, and we induce the dissipating state, which leads to aggregation of the antenna proteins. However, under enhanced gradient pH conditions, we have also the dissipating state in the absence of PSBS, which leads also to the aggregating state. There is, as I told you, an interplay between the presence of monogalactosyl deacylglycerol lipids and digalactosyl deacylglycerol lipids in the presence of PSBS. The first lipid is acting as a lateral pressure on the light harvesting complex to induce a conformational change and the dissipating state, while these lipids here in the presence of PSBS induce an hydrophobic mismatch. The thinning of the thylakoid membrane mismatches the hydrophobic region of the protein, which leads to aggregation of the antenna to overcome this mismatch. Now, we know that in the light harvesting conformation, there is a chlorophyll to chlorophyll energy transfer. Chlorophylls absorb energy from the sun, and the energy is hopping between chlorophylls with increased lifetime of the chlorophyll excited state. However, in photoprotective state, there is a disruption of the chlorophyll to chlorophyll energy transfer, and there is also a decreased lifetime of the chlorophyll excited states. In the literature, there is a special pair proposed to be the site of quenching. The pair is the chlorophyll A612 and the lutein 1 in this area here of the light harvesting complex. So instead of having chlorophyll energy transfer under light harvesting conditions, we have a chlorophyll to carotenoid energy transfer. To the carotenoid singlet short-lived states, so the energy is dissipated as heat. So how are our models doing on this basis? We have identified the light harvesting structure with an efficient chlorophyll energy transfer and a minimum interaction between chlorophyll 612 lutein 1, which means that we have no dissipation of energy as heat and increased lifetime of the chlorophyll excited states, which proves our point that this is the light harvesting state of the antenna protein. We also have experiments going on spinach leaves to prove our point. And some concluding remarks, molecular dynamics and with new sampling techniques and with the aid of high performance computing resources can lead to accurate description of the structure and function of the large protein complex at atomic scale, contributing essential knowledge on structural biology. Simulation outputs at atomic scale exert consistency with and can guide experiments with considerably lower costs. Over the last 2-3 years, we have obtained almost 50 million standard CPU core hours in 2 major projects from PRAISE. One is referring to COVID-19 and the other on photoprotection. And we have also aid from financial aid from Research and Innovation Foundation from Cyprus and for the human resources and the equipment for the experiments. This project, as I told you, is based on the clustering dynamics of the major light harvesting complex. It was a SuperMUC project under the PRAISE scheme. And we thank all the people involved in this. And also I thank my research team on photosynthesis. And thank you for your attention and the invitation to present my work. Thank you.